In this video, we will discuss about the biosynthesis of glycoRNA within the exosomes. So researchers basically confirm or check their hypothesis that the glycoRNAs are present in the exosomes or not. So, so these things they have confirmed by targeting three genes that are involved in this exosome process. First one is this RAS gene that involves in this ESR ESCRT dependent exosome process, and second one by targeting NSMASE that basically controls this ESCRT independent exosomal process, and the third one is by targeting this VSP or the ESCRT complex. So these are the three things that they use, and for targeting this ESCRT complex they use this um, or for targeting all three of these genes they use the short interfering RNAs method, and this is how they checked the mRNA formations in this HGS knockout, and they can see that the mRNA HGS mRNA's formations is less than 50%. Thereafter, they check the exosomal RNAs in those knockout conditions, not just HGS knockout, but also they use the inhibitors, these manumycins to reduce these ROS formations, and GW4869 to reduce this NS-MACE formation. So they knock down the HGS, but for RAS and NS-MACE they use the inhibitors. Let me clarify this thing. So in the exosomal RNAs, when they check the glycoRNAs, they are able to see reductions in this glycoRNAs when they use HGS knockout, and also when they use the inhibitors for RAS and inhibitors for NS-MACE, they see reductions in the glycoRNAs. In the combined inhibitors, they almost see no signals, and these things now they have validated in the quantification. They have quantified this Western blot in the panel D, where we can see that the signals for this glycoRNAs or signal of this IR800 stepped signals goes down in this HGS knockout, and when they use the MA or GW4869 to inhibit RAS and NS-MACE respectively. When they use both the inhibitors, they see further reductions in the IR800 streptavidin signals. Now, since the glycoRNAs are not coming out, so the hypothesis that these exosomes, since they are not able to secrete out of the cells, it will cause the accumulations of this exosome within the cells. So cellular RNAs, glyco signals in the cellular RNAs will increase. This is what their hypothesis. So when they check this thing, they are able to see a corresponding increase in the glyco RNAs within the cells, and this is true for both this HGS knockout, that is for this RNA1 targeting, and also for this RNA2 targeting. So in this case, so this is what their idea, and so what they have finally concluded is that when they knock down this RAS, this ESCRT complex will not be able to come out of the cells, and when they knock out this NS-MACE, and this ESCRT independent complex will not be able to knock out of the cells. So this will cause the accumulations of the glycoRNAs within the cells, and these things they have shown using this panel E, where they see a corresponding increase in the signals for the HGS knockdown, and here we can see that the signals are almost negligible in the exosomal RNAs, and these things they have quantified in the panel D. So from these results they finally come up to a point that if this biogenesis of the exosomes will be inhibited, then it will prevent the glycoRNAs to come out of the cells, and thus it will increase the glycoRNAs within the cells. Also, autocellular RNAs, glycosylated cellular RNAs increases. That's all for this video. If you need to understand more about their experiments in detail, you can go to the reference that has been provided in the description. Thank you for your time.